Good morning, everyone. My name is Yang Yang, and I'm from the University of Chicago, Benmei Department for Cancer Research, and I'm a part of Li Xinyang's lab. Today, I'm going to talk about one of my projects, large tandem duplication in cancer, result from transcription and the DNA replication collisions. So as we know that germline variants present in all cells and inherited, and the somatic variants specific to certain cell types and not inherited, and both possibly contribute to cancer development. And now there are many studies delve into the function of single nucleotide variants, SNV, structural variants, SVs, and the copy number variants, CNVs. And our lab focuses on the researching the mechanisms and the consequences of somatic structural variants. So structural variants are large genomic alterations, and usually they are detected by examining abnormal sequence assembling through whole genome sequencing. So for example, deletion refers to the loss of DNA segment. And the tandem duplication is one of a very popular popular structural variation involved to the repetition of a segment overall a very short distance. And the inversion represents the reversion of a segment. And the translocation refers to the connection between two different chromosomes. And the knowledge to the formation and the mechanism of structural variations are very limited. The only thing we know till now are that BRCA deficiency may lead to different kind of structural variation. For example, BRCA1 deficiency may lead to small tandem duplication, and the BRCA2 deficiency may lead to small deletions. So DNA replication stress is known to be a major source of genome instability. So we hypothesize that a subset of somatic structural variation in cancer may result from the DNA collisions, since DNA replication and transcription share the same DNA template. So it is unavoidable to cause a head-on collision or co-directional collisions. And this animation presents how this head-on collision happens. And we are asking what kind of structural variation may arise from the DNA collisions. So as different mechanisms may generate variations with different characteristics, I like this picture very much. This is a musical notation. So this process is very similar to picking out a melody, individual melody from a symphony, with each instrument contributing its unique tone. So non-negative matrix factorization is a very common use method to inferring and extracting signatures. And we already know that there are a bunch of point mutation signatures extracted now. And we know that SBS1 and SBS4 are associated with aging and smoking, respectively. However, the research on SV signatures remains relatively sparse. So our work is to do something with the structural variation signatures. And we performed this analysis in 3 large pan-cancer studies in several tumor cohorts. And since the results are pretty robust, we just show part of the data here. So we first classified the structural variations into 4 major types. They are deletions, tandem duplications, inversions, and translocations. And we further characterize them into different sizes from like 0 to 1 kb to like 10 kb. Then a total of 13 signatures were detected in the PCOG cohort. And there are 4 deletion-related signatures. From small to large, we name them deletion 1 to deletion 4. And also there are 4 tandem duplication signatures. From small to large, we name them TD1 to TD4. And also there are several signatures, for example, fragile site signatures, foldback inversion signatures, unbalanced inversion signatures, large mixed signatures and unbalanced translocation signatures. But I'm going to talk about the tandem duplication signatures today here. So a very similar signature profile was extracted from Hartwig metastatic tumor. So the peacock data was a primary tumor. And we also have a very similar profile extracted from the metastatic tumors. And there are two additional signatures, deletion 0 and TD0. They are extremely small SVs. And the reason why we can detect them is because maybe the SV is called by different algorithms. So it may be due to the differences in SV detection algorithms. So if the SV breakpoints are indeed have something to do with DNA replication, we expect to see the see the orientation of the breakpoints may be related to the DNA replication orientation. They may go with the unreplicated strand or go with the replicated strand. So to test the correlation between SV breakpoint and the DNA replication, we obtain the direction of DNA replication from consistent replic data from different tumor cell lines and get this consistent replication timing data. In this plot, this is replication timing data from MCF7 cell lines, and the peak of it is where the replication initiates, and the value of it is where the DNA replication ends. So from this plot, you can see the direction of the DNA replication. And to investigate whether there is a bias in the DNA replication direction, we randomly shuffle all the observed real SVs that fell within genes chromosome the genome. And by comparing the observed SVs with the shuffled and the randomly shuffled SVs, we found that the breakpoint of TD3 and TD4, which are large tandem duplication only, were consistently enriched in the replicated strand in both cohorts. So you can see here, only TD3 and TD4 are significant in the peacock and the Hartwig pan-cancer studies. So if large tandem duplication really result from transcription and the replication collision, we expected that the strand bias to be dependent on gene expression. So in unexpressed genes, we didn't observe any strand bias in TD3 or TD4, whereas the strand bias was clearly evident in expressed genes. And then we look at the number of large tandem duplications in different tumor types and samples. And the large of tandem duplication is pretty higher in female tumors like uterus, ovarian, breast cancer, and we also observe them a number of them in esophageal and stomach cancer. While I want to note that we noted that some of the patients, the prostate cancer patients, have extremely high frequency of large tandem duplications. And we also did the survival analysis, and it showed that higher large tandem duplication in tumors were associated with poor overall survival when all tumor types were combined in the peacock data, as well as when patients were stratified by TP53 mutation, because we know TP53 is always play some roles in the survival. And also large tandem duplications were also associated with poor survival in prostate cancer, skin melanoma, and esophageal cancers. Then we want to identify genes involved in the transcription and the replication collision and the large tandem duplication formation. So as expected, TP53 were positively associated with large tandem duplications, since we know that they allow the cells to tolerate genome instability. So besides the TP53, we found the CDK12 mutation associated with large tandem duplications in ovarian cancer and metastatic prostate cancer. And the function of CDK12 is known to control the transcription through phosphorylation of RNA polymerase II and regulates transcription, elongation, and termination. Therefore, we hypothesize that CDK12 inactivation can cause elevated transcription and replication collision. To test this hypothesis, we generate single-cell derived CDK12 knockout clones using CRISPR-Cas9 in two prostate cancer cell lines. One is C42B, one is 22RV1. And the loss of CDK12 significantly increased the total R loops. So the R loop is a biomarker of DNA replication and transcription collision. And we also performed a proximity ligation assay to measure the co-localization of RNA polymerase II and the DNA replication protein pCNA. And we found that CDK12 knockout cells had significantly increased the co-localization signals. And to directly assess the effect of CDK12 loss in genome instability, we culture the C42B cell line, which is also a prostate cancer cell line, both CDK12 knockout and control for a year to allow enough time for the somatic SV to form. And the whole genome sequencing was performed on the 6 single-cell expanded clones. And the parental cells were sequenced at a much higher coverage, 200x, so that the pre-existing SVs, if there were any, could be removed. And in the control clones, we found that there are like 63 to 155 somatic SVs were detected. But in the CDK12 knockout clones, there are 40 to 78 structural variations developed in 1 year. Although the type and the SVs numbers are varied among the knockout and the control clones, the fraction of large tandem duplication remained relatively stable. We can see that the CDK12 knockout clones had a significantly higher large tandem duplications than the control clones. And we also did a drug sensitivity test in 325 cell lines from CCLE database. And they revealed that cancer cell lines with abundant large tandem duplications were more sensitive to two drugs, two DNA repair-related drugs. One is WEE1 inhibitor, MK1775, and the other one is PARP inhibitor, PG34. And we did the drug dose response assays, also indicate that the loss of CDK12 significantly increased drug sensitivity to MK1775. So this suggests that the larger tandem duplication could be a biomarker for drug sensitivity. So here is our model. So we proposed this model to elucidate our observation. So in tumors, if the CDK12 is absent, the process of RNA polymerase II phosphorylation is disrupted and leading to the transcriptional dysregulation. And the dysregulated transcription results in the formation of R loops, and which subsequently block the DNA polymerase and the replication fork. And if these blockages are not properly repaired, they may result in DNA double-strand breaks and eventually lead to large tandem duplications. And there are still some of the questions are opening we haven't solved. For example, how in the large tandem duplication formed. And we are going to study it in our further study. So I'm going to thank my PI and lab mates from U Chicago. And also I want to thank our collaborators from Jonathan's lab in UC San Francisco for their support on the experiments. Okay. We have time to take any questions.
Okay, yeah. Yeah, thank you, Yang Yang. Let's have one quick question as Xiaoyu comes up. All right. Well, if there are no questions, then let's use this opportunity to maybe help get back on time a little bit. Um,